We have to talk about a few more things in terms of genetics. So this is part 2 of where we left off last time. Remember in the last lectures we were talking about homologous chromosomes, and this is in terms of autosomes. So chromosome sets 1 through 22. And if you look at the karyotype, we know 1 through 22 all have 2 copies of each chromosome, one that came from biological mother and one that came from the biological father. And on each of these homologous chromosomes, there's alleles, many different alleles, many different versions of the same gene, but one that you inherited from your biological mother and one from your father. And we talked about that there are some autosomal disorders, which means there's a genetic defect on any of the chromosomes from 1 to 22. But now we have to talk about the last set of chromosomes, which are the sex chromosomes, X or Y. This is a really unique case because X and Y are not homologous at all. They're very different from one another. As we know, all eggs contain an X. All sperm either contain an X or a Y. So there's a 50-50 chance that a person could be either XX or XY. But no matter what, notice that everybody has at least one X. It's something that every person has in common. The X is substantially larger than the Y. The Y chromosome is very small, and there's really not a lot going on in the Y chromosome. There's not very many genes on it. The X, though, is huge, and it has tons of genes. That's why whenever you see these karyotypes, the Y is always so small because it's, doesn't have a lot of things going on it. The X is huge because there's so many genes on the X chromosome. Actually, if you zoom in and look at the genes on the X or Y chromosome, the Y really only has 2 really important gene segments. One of these is called the SRY region. This is the sex determination region. These genes code for things that allow for development of testes, and also these genes in the SRY region prohibit the development of uterus and fallopian tubes and other female reproductive structures in an XY male. So that's one region in the Y chromosome that's different. The other one is this AZF gene region, which codes for sperm development. At the same time, like I said, the X chromosome is huge and has a lot of genes. So what happens when one of these genes is mutated? Like, let's say that we had somebody who had 2 X chromosomes compared to somebody who only had 1. So in this drawing, these are the 2 X chromosomes. This is an X and this is a Y. Let's say that this is one of the alleles that we're talking about. All right, and normally it's green here. This is the normal allele, and it's found only on the X. But let's say if this normal allele is purple, this is a mutated form of the allele. Well, remember, if somebody is XX, they have 2 X chromosomes. Normally, the normal allele is dominant over the mutated form. So if this were a case where there's a mutated allele on one chromosome, this normal one would mask the mutated form. So basically, this person may be a carrier of a disorder because they have one mutated allele, but their other one masks the mutation. On the other hand, if somebody's XY and they have a mutated form of the allele, remember the Y doesn't have anything on it, so there's nothing there to protect this mutated X. So therefore, if somebody has this mutated allele and they have only one X chromosome, they now have the disorder. They're not just a carrier. So any disorder that can be carried on this X chromosome is called a sex-linked disorder. And like I mentioned, sex-linked disorders are almost always on the X chromosome only, and they're almost always recessive, meaning if somebody has a normal version, it'll mask the mutated version. This picture is showing several examples of sex-linked disorders that could be caused due to a mutation on the X chromosome. I'll let you guys look some of these up if you're interested. Okay, ocular albinism, muscular dystrophy, androgen insensitivity, SCID, which is severe combined immunodeficiency disorder, hemophilia, which means the blood doesn't clot properly, and red-green color blindness. All of these are disorders that could be due to mutations on the X chromosome. So let's do an example of one of these X-linked disorder problems. So let's say that the allele we're testing for is the B allele, which codes for either normal vision, which is dominant, or color blindness, which is recessive. Now, since these alleles are found on X chromosomes, chromosomes, the way that you would write this is you would write an X. And if you had an X with a big B, this indicates, again, normal vision. But if you have an X with a little b, this is the color blindness. So when you're writing these X-linked alleles, you don't write them just like this. You have to write the X with either the big B or the little b. So let's say we had an individual who was XY, and then we had another individual who was XX. Now let's say this XY individual had normal vision. What we would do is we would put that big B here next to the X, but the Y does not carry an allele on it. Remember, the Y does not have all of these alleles, so the Y doesn't get anything. You don't put any B next to the Y. Now let's say this other individual who was XX was heterozygous and they were a carrier of color blindness. So what this would mean is they had one X with a big B and one X with a little b. Even though they're a carrier, they still have normal vision because they have one dominant allele. Now let's say that these two were going to cross with one another. You would draw your Punnett square like always. Now what you have to do, since this is an XY scenario, is one of these boxes will get the X, the other one will get the Y, and then it's crossing with this XX individual, so these would get X's. And now you have to put the B's. So the XY individual had a big B next to their X, and remember the Y doesn't get anything. The XX individual had one big B and one little b. And now you're going to cross them like you always do. So now, when you look at the results, what you can see is that 2 out of 4 of their potential offspring are XX. So there's a 50% chance that their offspring will have XX, and there's a 50% chance that their offspring may be XY. We got that part down, no problem. Now we have to look at other things. For example, what are the chances of them having an XX offspring who has normal vision? What are the chances the XX offspring will be colorblind? And what are the chances they'll be a carrier? So if we look at this, both of the XX possibilities have one big B. That tells you normal vision. So all of the possible XX offspring will have normal vision, 100%. Will any of the XX offspring be colorblind? The answer is definitely not, because in order to be colorblind, the offspring would have to be XX with 2 little b's, but there are none, so there's 0% chance. What is the chance of the XX offspring being a carrier? Well, there's 1 here, 1 out of the 2. So out of the XX individuals, 1 out of 2 of the XX will be a carrier. Now let's look at the XYs. What are the chances of the XY having normal vision? Well, 1 out of the 2 possible XYs will have normal vision. So 1 out of the 2 XYs, or 50%, will have normal vision. What are the chances of having an XY offspring who's colorblind? Again, 1 out of the 2, 50%. What are the chances of the XY being a carrier? We wouldn't say this. We would say this is not possible because, remember, an XY individual only has 1 copy of the X. So they really couldn't be a carrier. It's either they have the disorder or they don't. So that's how you would do a problem like this. And as you can see, it's much more frequent that an XY individual would have an X-linked disorder than an XX individual. Hey, this is a pedigree showing again what I was talking about, how it often skips a generation. See, look at how this grandfather was colorblind, but their son here is not colorblind. Whereas if he had a daughter who was a carrier of colorblindness, it could be very possible that this daughter could spread it to her son. So it skipped a generation. That's often what happens with things like colorblindness or hemophilia and all these other sex-linked disorders. Hey, here's another problem we should do together. Okay, a father, so that's going to be a square, with hemophilia. It's a recessive X-linked condition. So we can write up here, X with a little h will be hemophilia, and X with a big H will be normal blood clotting. This father is going to be X with a little h and Y, and somewhere he mated with this XX, but we don't know what kind of hemophilia genes this mother had. So this pair is going to have a daughter who is not affected by hemophilia. So we know this daughter must be XX. We know that she inherited at least 1 little h, but if she's not affected with hemophilia, we must know that she's a carrier. The other one must have a big H. The daughter will mate with an unaffected man. So again, we're going to draw a square. We know this individual is not affected, so they must have a big H here. What is the probability that a daughter of this mating will be hemophiliac? So we have to make a square. We can put the sperm possibilities at the top, so X with a big H or a Y. We'll put the egg possibilities on the side, X with a little h or X with a big H. And now we'll cross them. So what is the probability that this will result in a daughter with hemophilia? Well, clearly it's 0. However, it's possible that 1 out of 2 of them would be a carrier. On the other hand, what is the probability of this pairing having a son who is a hemophiliac? Well, we see this one has the small h, so we know there's a 50% chance of having a hemophiliac son. But there's also a 50% chance of having a son with a normal blood clotting phenotype. So that's how you would do these kind of X-linked condition problems.